Thank you very much. Can you hear me all right? Can see everything? Is that
Yes, we
okay?
can. Yes, we can.
Fantastic. So first of all, thank you, Muhammad, for the fantastic organization. Thank you, Naboza, for the very kind and nice introduction. It's a great pleasure to be back in Oxford, though it's only in a virtual mode. I really hope we'll get over this pandemic and that I can come back to Oxford in person. So today, as Nabuza said, I would talk a bit about what we do with these agents here, the modified nucleoside triphosphate. And in particular, I will present two main research directions we follow in my lab. The first is, and this has been beautifully introduced by Amy just in the previous talk, we use CellX with these modified nucleoside triphosphate to try to identify chemically modified functional nucleic acids, in particular aptamers. We also have really started to use these modified nucleoside triphosphate in something called controlled enzymatic synthesis, which we hope to develop as a valid alternative to phosphoramidite chemistry for the synthesis of DNA and modified oligonucleotides. So let me start with first our involvement in CellX. So I think I do not really need to go into many details, but the main idea here is to use to synthesize modified nucleoside triphosphate, use them in CellX, and get modified aptamers for various applications. So in general, when people identify aptamers, they use non-modified DNA or RNA, and for that they use libraries to generate these libraries. First of all, you use phosphoramidite chemistry to get the libraries and then amplify them using PCR. And in the lab, we pursue two um, research axis. First of all, we a bit like what they do at Somamer, at SomaLogic, but like we're not as efficient. But we replace one or multiple nucleotides with chemically modified counterparts, and we call these exotic functional nucleic acid functional groups. And I'll get back to that in a minute. In the second strategy, we try to expand the genetic alphabet by having, in addition to the ACGT base pairs, an additional XY base pair that is mediated by metal binding. So in essence, we ask polymerases to incorporate a modified nucleotide Y strictly and orthogonally opposite an X nucleotide on the template strand in the presence of metal cations. This would obviously enable to increase the chemical diversity that would be available for selects in general. So we'll present to you a bit what we do in these two research directions. So when I started my lab and about 6 years ago now, um, I knew that the synthesis of these modified nucleoside triphosphates is not always a very easy undertaking. So we uh, decided to try to use a chemical synthon here, EDU, TP, that could be used to generate a small subset library of modified nucleotides by a simple click chemistry using various azide modifications. Once established, this library then can be used in CellX to get to modified aptamers. Since then, we've synthesized quite a few of these modified nucleotides with various side chains, which are totally alien to what is found in nature. So here we have a cubane moiety, and I will talk about that in more details. We have also recently synthesized one that has a vancomycin on it and other functional groups. So to exemplify this strategy, I'll just briefly mention a specific modification, which is called the cubane. So the cubane, as the name says, is a hydrocarbon consists of 8 carbon atoms that are connected together to adopt the shape of a cube. It's an isoster of benzene, meaning that it shares a number of physicochemical properties with benzene. It has obviously also differences with benzene. First of all, it's not planar, it's not aromatic. there is a decent, an important ring strain in the cubane, making all the hydrogen atoms quite acidic, which in turn makes them engage into non-classical CHO hydrogen bonding interactions. Interestingly, also, um, cubane has attracted a lot of interest in medicinal chemistry because there was a publication about 6 years ago where they replaced phenyl and aromatic rings of drugs and replaced them with cubane moieties, and showed actually that these uh, new drugs were equally potent or more potent than the original versions. So obviously we were very interested by this modification in the context of aptamer selection. So we synthesized the corresponding modified nucleotide, showed its compatibility with enzymatic DNA synthesis. And then together with a colleague in Hong Kong, Julian Tanner, we then did a select experiment against the protein PVLDH, which is a lactate dehydrogenase from PVVAX, and is an excellent biomarker for malaria. We're lucky enough to identify various sequences. We synthesize them using solid phase chemistry, and tested all these sequences, and the most abundant was also the best binder. While the KD values are not stellar, the advantage of this cubomer is that it is binding only to the LDH from PVVAX and not to the one from P falciparum, despite an over 90% sequence homology, which is remarkable. We also resolved the crystal structure of the aptamer protein complex, and with a 2.5 angstrom resolution. And I would like to pinpoint to highlight two structure key features which have also been identified in quite a few of the somomers. Actually, there are four cubane residues that are forming sort of a hydrophobic cubane cluster, which inserts into a hydrophobic pocket of the PVLDH protein. And we believe that this is the main mechanism that mediates binding and interaction between the aptamer and the protein. There's another interesting structure key feature. One of these cubane residues is in perfect positioning, that is in terms of distance and bond angle, with a carbonyl moiety of the backbone of one of the amino acids of the protein, and forms what we believe is one of these CHO bonding interactions. And we believe that this unusual CHO bonding is crucial for specificity and protein recognition. Our collaborators in Hong Kong then used the cubomer in a mimetic clinical situation to identify and distinguish PVLDH from PFLDH and successfully worked in two different types of assays. Now, for the metal-based approach, this is something that we have started a few years ago, and this will be a long-term project because it's not as easy as it seems. So the idea here is to have artificial nucleotides that would act as ligands for metal coordination. The idea is that obviously these would be orthogonal to canonical nucleotides, which allow for an expansion of the genetic alphabet, and might solve some of the drawbacks involved in hydrophobic aromatic artificial metal base pairs that have been reported, for instance, by the group of Romsberg and Hirao and Steve Benner in the States. Now, it's not as easy as it seems because we ask a lot from the system. So first of all, we have to synthesize the modified nucleotides, have also modified templates, and then screen polymerases to incorporate specifically the modified nucleotide opposite the modified templating nucleotide in the strict presence of metal cations that we have to screen also. So you can see that this is not particularly easy. And once installed, actually, and this is probably one of the major um, hurdles in that context, is to find uh, enzymes or a metal base pair that does not block DNA synthesis, enzymatic DNA synthesis. And this is also something that has been noticed with other artificial base pairs. So our best candidates so far are these two. So one is sort of an imidazole modified nucleotides, where the nucleobase was replaced by this imidazole moiety that bears a carbonyl function to further coordinate metal cations, and this modified purine nucleotide that has a pyridine instead of the amine of adenine, to sort of have a bipyridine system. The use of these two modified nucleotides is quite convenient and works decently well, because we can form with high efficiency silver mediated artificial metal base pairs. And even more, actually, once installed, we can continue DNA synthesis with unmodified nucleotides. However, the problem with these modified nucleotides is that there is partial incorporation opposite natural nucleotides, which makes the system not fully orthogonal, and for the time being cannot be used in selects experiments. We're continuously screening other nucleotides, and hopefully we'll find better candidates. Recently, we have also expanded that to not only DNA, but also RNA nucleotides, with a very simple system shown here. It's just the incorporation of ribouridine nucleotides opposite templating DNA DT by mercury-mediated metal base pairs. And we could show that actually, first of all, it forms stable metal base pairs, but also that they can be used for the synthesis of at least two modified base pair, which opens up the possibility of having different chemistries than DNA at the level of the sugar in this context. Now, let me finish this presentation by summarizing our recent results that we have obtained in the field of controlled enzymatic synthesis. So first of all, what is controlled enzymatic synthesis? So you all know that most RNA and DNA oligonucleotides can be obtained by solid phase synthesis, shown here. I'm not going to go through the details, but essentially it uses phosphoramidite chemistry. While this is obviously a very convenient and versatile and robust method for the synthesis of these oligonucleotides, um, it also permits the introduction of a lot of chemical modifications at all levels of the nucleosidic scaffold, be it at the level of the sugar, the phosphate backbone, or also the nucleobases. It even allows for the production of large-scale production of oligonucleotides. We're talking here at the kilogram scale level. On the other hand, this method is restricted to rather short sequences, that is up to 110 nucleotides in length. Also, not all functional groups are tolerated by the rather harsh conditions that are used during synthesis. And obviously, being a chemical method rather than an enzymatic method, there is no possibility for amplification. And the last limitation is that the number of oligonucleotides that can be synthesized at the same spot is rather limited to maybe 10, 20, 30, no more than that. Whereas like if you had enzymatic synthesis, you could produce much more. So in controlled enzymatic synthesis, the idea is to sort of combine solid phase synthesis with enzymatic synthesis. The basic concept is very straightforward. So the idea is to have a modified nucleotide with a protecting group, a temporary blocking group at 3' position, use a polymerase, usually the terminal deoxynucleotide transferase, TDT, which appends nucleotides on single-stranded DNA oligonucleotides. Obviously, the 3' blocking group will prevent the addition of another incoming nucleotide. And at that stage, the TDT and the DNTP are removed, and the blocking group is removed by deblocking conditions, and the resulting DNA strand is then ready for a new cycle. Now, this on paper looks very straightforward and easy, and there have been a decent progress made, especially by companies such as DNA Script, but we're asking quite a lot of the system. So we have to identify 3' temporary blocking groups, and these must satisfy a number of criteria. First of all, there needs to be a fine balance between stability versus ease of deprotection. So it needs to be stable enough so that it doesn't fall off during the enzymatic reaction or during storage. And at the same time, it needs to be easily removed so that this step here does not become a pitfall. Also, the protecting group cannot be too bulky, because otherwise it will not be accepted by polymerases anymore, and the single incorporation events need to be high yielding. So in one recent example, we have focused on the use of the 3' phosphate as a protecting group. So the idea here is to synthesize a nucleotide that has a 3' phosphate moiety and obviously a 5' triphosphate moiety. The incorporation by polymerases obviously would lead to a 3' blocked, phosphate blocked sequence that will avoid the incorporation of additional nucleotides. The use of a phosphatase would then remove the phosphate group, and then would resume the cycle using this strategy. So this looked like a very good idea, and preliminary tests actually, where we use phosphorylated primers in prime extension reactions and PCR, actually showed that using 3' phosphorylated primers blocked DNA synthesis, and then using a phosphatase such as the FAST-AP thermosensitive alkaline phosphatase allowed to restore DNA synthesis. So this looked very good. So we synthesized actually these modified nucleotides using new synthetic routes, and started to incorporate these modified nucleotides. However, to our surprise, we observed that actually not only one, but multiple nucleotides were incorporated, which was very surprising at the time. So we did an LCMS analysis, and actually this analysis showed that natural nucleotides such as the ATP were preferentially misincorporated rather than using these phosphorylated nucleotides. And this explained why we had this longer than expected products that were formed. So we then changed to a different system to better understand all this, and used a template that has a 7A overhang, and used these modified nucleotides. And even there, we observed that multiple incorporation events proceeded, and actually LCMS analysis confirmed that multiple T-LNA or natural nucleotides were incorporated. And to rationalize these findings, we synthesized then two nucleotide analogs, one that has a beta-cyano moiety appended on the phosphate group to probe the effect of charge on the phosphate moiety, and the one that has a phosphorothioate moiety on the 3' phosphate to evaluate the effect of hydrolysis. A long story made short, actually, we identified that the number of polymerases were actually capable of hydrolyzing phosphate moiety appended at the 3' position of incoming nucleotides, and thus had an unexpected phosphatase activity, which shows that actually the design of such nucleotides is even more challenging than initially believed. In the course of these experiments, and this is going to be my last scientific slide, actually, we also made this phosphorothioate containing LNA nucleotide. Um, and we actually could show that it was a substrate for polymerases, though there was a stereo preference for the S enantiomer um, rather than for the S diastereomer, which is not the case for the canonical nucleotide, which is readily incorporated into DNA. We also showed that actually this nucleotide can be used for the synthesis of modified um, nucleotides containing these dual modification patterns using standard primer extension reactions. So I hope that in this presentation, I could show you a bit what we tried to do with modified nucleotides. Mainly, we use them in selects to identify functional nucleic acids with enhanced properties. And we have started a program on trying to develop controlled enzymatic synthesis of DNA, RNA, and XNAs. All this work would not have been possible without the fantastic team. I would like to especially acknowledge postdoc Pascal Rutlisberger, who did all the selects experiments on the Cuban moiety together with research engineer Fabien Levy-Acobas. A lot of the work on the enzymatic, controlled enzymatic synthesis was done by postdoc Marie Flamme. I'd like to thank all my collaborators at the Institute and all my collaborators outside the Institute, especially Julian Tanner at the University of Hong Kong, where we have done this beautiful work on the cubomer. We'd like to thank all the funding that we had over the last years, and I would like to thank you very much again for the kind invitation and all of you for your kind attention. Very happy to take up any questions. Thanks a lot.